We're still missing one person. Yeah, now we are complete. Welcome to the official PhD defense of Ania Fiksinski. Professor Knape will be the chairperson today, and I give now the word to Professor Knape.
Thank you very much, Madam Beadle. Ladies and gentlemen, most welcome to the promotion ceremony of Ania Fiksinski. I have to apologize that you see another chairman than the chairman you were expecting. Professor Wokke unfortunately fell ill yesterday, and I have the honor and privilege to replace him, and I will do my utmost to make it a very nice and stylish ceremony, because we owe that to the promovenda who has been working very hard in the last few years. I hope that a lot of people are following the live stream and can attend this meeting. For those who are not quite familiar with promotion ceremonies, I would like to say that this is not going to be an exam. For an exam, you have to study hard. That is what the promovenda has done, of course, but she has also worked hard. She has prepared a manuscript which has been evaluated by the evaluation committee and has been approved. And what we would like to see is whether the candidate will be able, in the next 45 minutes, to conduct a scientific interview on a high level with the members of this committee who are all expert in the field, but who are also, moreover, very interested in the work of the promovenda. So before we start, I would like to introduce a few people. First of all, myself. My name is Hans Knape. I am the deputy rector. I would like to introduce the promoters, Professor Kahn, Professor Bassett, and co-promoter Dr. Forstman. And I would like to introduce a few members of the promotion committee who will not be going to discuss with the candidate. That is Professor Wiepke Kahn, Professor Kastelein, Professor Mink van der Molen, and Professor Schepers. Madam candidate, are you ready?
I am.
You're ready. Your paronyms, are they ready as well? They are. They are. Thank you very much. Well, let us start. I hereby open this session. Madam Doctoral Candidate, the Board for the Conferral of Doctoral Degrees of Utrecht University now grants you the opportunity to defend your public dissertation in public. And I invite the first opponent to speak. And the first opponent will be Professor Wijnen. He is Professor of Psycholinguistics here at Utrecht University, and moreover, he is the Chair of the Evaluation Committee. And he has got the word. Please go ahead, Professor Wijnen.
Thank you very much, Mr. Chairman. Dear Madam Candidate, let me start off by complimenting you on having completed and congratulating you on having completed a very fine piece of work. It's an impressive book that you produced, a number of very well-designed and well-reported studies. It was a joy to read, and it was also very instructive. Obviously, I'm not here to only praise you, but also to discuss with you some of the aspects that I found maybe a little less convincing. One of those issues is, actually there were very few, let me stress that. One of those issues has to do with, is actually in chapter 4, in which you measure the abilities of a group of people with and without 22q11 deletion syndrome on various neurocognitive domains, and you use these measurements to predict outcomes using the Vineland adaptive behavioral scales. When I was looking at your figure 1 and the tables 1 and 2, I was a bit puzzled by some of the outcomes of the principal component analysis that you conducted. And let me just illustrate what I mean. First of all, there is a cluster of items, and obviously the figures, the statistics tell us that this is a cluster, but there is something strange about it anyway, I think, which is called visual and logical memory. And I have no idea what the concept or construct of visual and logical memory might mean. When I look at the items that load on this component, I see that there are subtests. The Wechsler's memory scale for logical relations, I think it's called. That's to do with the recall of concepts that have been presented in the context of an orally presented story. So I wondered how you could connect that with visual memory, for instance, or visual skills. Another example is the judgment of line orientation, which I found to be loading mostly and clustered under component 4, which you call executive performance. Visual reproduction 1 is also loading on component 4. And truly, the loading factor is higher than on component 1, but nonetheless, I would feel inclined to cluster it with component 1 rather than component 4. There doesn't seem to be a substantive connection between visual reproduction and executive performance, but maybe I'm mistaken. So these kinds of observations make me wonder what's going on there. Obviously, again, as I already stated, the statistics tell us one thing, but looking into what these tests apparently are doing may tell us a different thing. So does this tell us anything about, for instance, the purity of the tests or the validity of the tests that you've been using? Does it tell us something about the principal component analysis you've been conducting or something else? So please help me out here.
Hooggeleerde opponent, let me start by thanking you very much for your kind and positive words about my work. With your question, you're referring to the work presented in chapter 4, where I investigated a cohort of adult individuals with the 22q11.2 deletion syndrome, both with and without schizophrenia spectrum disorder. And we tested them for their neurocognitive functioning and tested whether there was an association between domains of neurocognitive functioning and subsequent daily life adaptive functioning. And you are commenting on the validity and basically you're questioning these principal components, so the domains of neurocognitive functioning. And I am actually glad that you point this out because this has been a very important topic of discussion amongst me and my co-authors on this study. So let me start by saying that what happened is that we conducted or we assessed 15 different very specific and minor, one could say, neurocognitive tests in all these individuals. And then we used, as you mentioned, a data-driven approach to see what domains would arise. Now, I completely agree with you that this is not ideal. However, I would argue that it is better than the alternative, which would be to use solely a theory-driven approach to cluster these specific neurocognitive tests.
Why would you?
Yes.
Why do you think so?
I would argue that the major drawback of using only a theory-driven approach to doing so is that these tests are very specific, and it's a known issue in psychological research that their validity and what's the word I'm looking for? That they do not necessarily capture one overlying construct that you can specify by theory, and this is why we opted to use a data-driven approach. Thinking our rationale for this was that let the data tell us what components cluster together, and so to sum this up, because it's a little bit of a technical story, I think, is that I agree with your comment that it is not optimal to use a data-driven approach to do this, but I do argue that it is better, particularly going forward. If one would have a larger sample size, one could even further improve this method. And now, with your question, you're also referring to a few specifics. Mm-hmm. One example that you mentioned is the construct of visual logical memory. This is actually also one of the examples that we pointed out as it's striking what specific tests load on this domain, and one could question their validity indeed. So it's something that we agree on, I think. When we thought about this, an important argument was, or an important hypothesis, one could say, was that we believed that the underlying mechanism is probably that the verbal memory retention tasks are very much visually stored in the brain, and so therefore might represent an overall construct of a visual and logical memory, as opposed to the more concrete tasks that belong to the verbal memory domain. And
could you comment? Yeah, I see that point. I mean, it is largely speculative, of course.
Yes.
But could you also comment on these line orientation tasks, which I found particularly intriguing?
Yeah, so the judgment of line orientation task loads most strongly on the component of executive performance, indeed. Right. Well, I'm actually, this is surprising, but I didn't find this result the most surprising, to be honest, because it's of course, it's a visual task. So this judgment of line orientation task requires one to compare various lines and their literal orientation, and to then pinpoint the correct corresponding line, basically. And this has something to do with, with the overall skill that that we use for what we would call executive functioning. So it's very much of a, while it is a visual task, it is not the case that it is a visual memory task or a visual motor coordination task. It is really something that relies on how you can store a construct in your brain and then can make the connection to the stimulus.
I'm afraid that we are coming to the end of this discussion, because
I'm fully satisfied. Very good answer. Thank you.
That is quite convenient. Well, you've had your first opponent, dear promovenda, dear Ms. Fiksinski, and the first opponent is usually the most difficult ones, but you did it quite well, so it gives a lot of hope for the next. We continue with the next opponent, and as you have seen, your thesis has not only aroused interest in Utrecht, but also in other universities, both nationally and internationally. And we go therefore to Maastricht University to meet Professor van Amersfoort, who is Professor of Translational Psychiatry, and she will continue the opposition. Please go ahead.
Thank you. Dear Madam Candidate, also I want to congratulate you on this beautiful, well-written thesis, and I also extend my congratulations to your supervising team. And your thesis consists of 6 nice papers that almost all have been published already in high-impact journals. And I'm particularly impressed that you make use of a unique data set and approach it from so many different angles. You're one of the few, I think, in this field who study both children and adults, and you used longitudinal cross-sectional methods. You approached the data categorically and dimensionally, and so yes, that's very nice. However, that makes it also difficult to draw one firm conclusion, I think after reading your thesis, I had even more questions than I had before. But that's all what science about. I want to discuss with you about chapter 3, and that's a chapter in which you study children with 22q11 deletion syndrome from the Dutch sample, and you reassess them a couple of years later, approximately 5 years later, and you look at the risk of developing psychotic symptoms or psychotic disorder in those who have autism and those who don't. And I was surprised by your conclusions, and I wonder if you were as well, because from the general population literature, we know that people with autism at a young age have an increased risk of developing psychotic disorder in adulthood, and that's been even studied by our Dutch colleague Jean-Paul Celte, and it has been replicated in several samples. And also there's enough indication for shared genetic liability. So how do you explain in this background your findings of no increased risk and no association between these two?
Hooggeleerde opponent. Thank you very much for your kind words. Your question refers to our study of children with 22q, where we investigated the association between autism spectrum and subsequent psychosis risk. And I understand your surprise with respect to the results, because what we find is that there was no positive association between autism symptoms and subsequent psychosis risk. While, as you point out, in the general population, these two phenotypes do co-occur at increased rates. I was not as surprised, I think, as you may be suggesting, and that has actually a very basic reason, and that is, I think, my modest clinical experience or observations, because these are all children that we have, well, that I have assessed together with my colleagues, and it was not my impression, at least, that those children with an autism spectrum diagnosis or autistic symptoms were the ones that then a few years later I would see them back and they would have developed a psychotic disorder. So that is perhaps a background to the lack of my surprise. But then let's return to the study and to the theory. An important reason why, or I think what these results point to, is that an important reason why there is co-occurrence of ASD and psychosis in the general population is that they indeed, as you say, there are many genetic etiological factors that may predispose the risk for both. However, as this study points out, and as we now increasingly know from the other literature that is emerging in this subject, not only in the context of 22q, but also in the context of other pathogenic variants, the concept of pleiotropy is rule rather than exception in the context of a pathogenic variant such as 22q. So with pleiotropy, I mean to refer to the fact that in this case, one...
Sorry to interrupt you, but I understand your concept of pleiotropy. But I still am not completely satisfied, because why did you assess these kids for psychosis at the age of 19? Was that the most logical time point? The development of psychotic disorder, would you have expected different results if you would have waited in line with the IBBC protocol a couple of years later?
Yeah. Well, with this question, you point out a potential limitation of our study, namely, indeed, that the last age of assessment was 19, whereas we would want to consider the age up until the age of 25 to be more certain to say whether someone will truly not develop schizophrenia, or at least the chances will be far less likely. However, I do think it's important that we assess these children also at age 19 and even younger for the presence of a psychotic illness or symptoms, because we do know that they do occur also in younger children, adolescents, and young adults with and without schizophrenia.
But are you planning to follow them up? Because wouldn't it feel like it's unfinished work here?
Yes. So yes, we are in fact following them up as we speak. So we do invite them back after another 2-3 years, at least until they're 25 years old. That's what we would like to do. And what I would also like to add to my answer is that we thought of this limitation, of course, and we thought maybe we could check whether, they are not at the age where we can say for certain whether they have developed schizophrenia or not. So let's also look at subthreshold psychotic symptoms that we know may be a prodromal phase of a schizophrenia illness expression. So besides looking at psychotic illness as an outcome factor, we also looked at psychotic symptoms. And
I want to ask you one more thing before my time is up on this chapter. Could it have something to do with the fact that your autism diagnosis were mainly PDD-NOS, PDD-NOS? What would happen if you would apply the current DSM-5 measures? Would you have the same results, you think?
Please give a short answer, Dr. Ms. Fiksinski.
Yes, I do think we would get the same results. That's
Why? I'm not convinced.
Yes, well, our and other studies support the fact that the heterogeneity that we observe and the dimensionality in the autism phenotype is not specific to autism within 22q, but is rather general, is a general truth for this phenotype across the board, not only in 22q, but also in idiopathic autism populations.
Okay, thank you. I think my time is up. Thank you very much, and I pass the word back to the director.
Thank you, Professor van Amersfoort. As you mentioned in the beginning, this thesis raises more questions than answers, certainly than can be given within these 45 minutes. We have returned to Utrecht to meet Professor Burbach. Professor Burbach is Professor of Translational Neuroscience. Professor Burbach, please go ahead.
Thank you, Mr. Chairman. Waarde promovenda, dear Madam Candidate, it's a real honor to discuss with you your thesis. I was really impressed by your thesis, not only by the profound and robust studies, but also the way you were able to present it to people that are really in different fields like myself. What I really liked were the problems that you posed in the introduction, very specifically defined and numbered, and you come back to that in the discussion. And to me, it was a great help to get a grip on the extent of your studies. I would appreciate to discuss with you more the genotype-phenotype relationships that are underlying the studies that you're doing. And for instance, in chapter 7, you work with a cohort. And as you know, there are different forms of the 20q11 deletions, short and long forms. And I think the forms are not represented in an equal way, although for schizophrenia it doesn't make a difference. But how about all the other phenotypes that you're studying? Can you comment on that? How about cognitive decline or IQ? Or perhaps autistic-like features, are they also, let's say, equally homogeneous if you have these heterogeneity in this region?
Hooggeleerde opponent, thank you very much for your kind words. Let me note that specifying these 4 problems and structuring my work according to them was helpful to me as well. So I'm glad you found them helpful too. With your comment or your question, you're asking me about genotype-phenotype associations, and specifically, I think you're asking what I can say about the association of deletion size or heterogeneity of the deletion size and type with respect to phenotypic outcomes.
Yeah, and particularly it's a concern because it may, let's say, provide noise or heterogeneity in your data set that you like to avoid.
Yeah. I understand. And as you point out yourself, there has been no association reported between the deletion size and presence of schizophrenia. I think, let me start by saying something more general. It has been a very intuitive hypothesis to investigate and to question whether, okay, we see all these individuals with the same deletion syndrome, but we see such heterogeneity in their phenotypic outcomes. So it seems that it seems very reasonable to expect that factors of this deletion size, type, specific modulating or other regulatory genes within the region, that they may be specifically associated to the heterogeneity. However, I think the fairest conclusion is that decades of research have not revealed a one-on-one association between deletion type or any specific aspects of it to any particular phenotype. And
is that also true for, let's say, the peripheral phenotypes like scoliosis, schisis, heart defects, etc.?
Yeah. Well, by making the disclaimer that is not necessarily my area of expertise, I do know that there are associations reported, for example, between the gene TBX1 and congenital heart defects. And also I wanted to point out something that is a little bit more closely related to our work, or to this chapter also, is that IQ, so cognitive outcomes, of course, are a major phenotype of interest in my work. And there has been one study that actually does report that a very tentative association that a smaller deletion, so the deletion, the typical deletion is between breakpoints A and D, and that a smaller deletion between breakpoints A and B may result in a slightly milder cognitive phenotype than the full deletion. However, it's I think we need to be cautious in interpreting this as of yet.
Yeah.
And
okay. I think you are not really worried about it. But before continuing this aspect, perhaps since I have still 1 minute left, I was also curious about a totally different aspect, and that is there is another study, Kleinen et al. In Nature Medicine. There's certainly an overlap, both in patient cohorts and readouts. Can you explain to me very briefly what is the actual highlight of your own paper in this thesis with respect to Kleinen? And
yes.
I noted that Kleinen et al. Wasn't referenced completely, which I was surprised about, but that's a side point. So what is your point about nature medicine as compared to molecular psychiatry?
Okay. Well, first of all, your side point, I will definitely see to that, because that's surprising to me as well. The highlight of our Nature Medicine paper, or the advance of our Nature Medicine paper over the Kleinen et al. Paper, is that we included other phenotypes and had a slightly larger sample. So while the Kleinen et al. Paper really focused on the schizophrenia outcome as the main phenotype of interest, we included what we called early schizophrenia-associated phenotypes, specifically IQ, baseline IQ, but also the phenotype of cognitive decline and the presence of subthreshold psychotic symptoms, because we were interested to not only identify an association, but also potentially get some more insight into the disease mechanisms.
Yeah, yeah.
So that's the first advance. And the second advance that I would like to mention is that we have started thinking about and working towards a method where we can actually use information from the rest of the genome summarized in a polygenic score or a polygenic risk score.
But there's also an important part that you left out in your paper, and that is the comparison to the Kirov gene set that is quite prominent in the Kleinen paper, but is not addressed in your paper. Is that still something you would like to do? You can just give me a yes or no, because my time is up. Will you look further into the genotypes that are involved?
Yes.
Okay, well, I'm happy about that. So
yes.
Thank you for your for your answers, and I return the word to the rector. Thank you.
Well done, Professor Burba. Thank you for sticking to the time. For the next opponent, we move to the western part of the Netherlands, to Leiden University, and we are quite proud that Leiden University has sent one of his experts in the person of Professor Staal. Professor Staal is professor of child psychiatry here in Leiden, and he will continue the discussion. Please, Professor Staal.
Thank you, Mr. Rector. Dear Madam PhD student, now promovenda, I would like to congratulate you as well. And of course, I could repeat what all the others have said, but I would like to try it a bit more original. I think it's a good example of talent being brought in the right environment and what it can produce for science. It's impressive work, and I would like to go to the more general scope of your thesis in the questions I have. You have in the summary a nice chapter that goes about, or paragraph I should say, about the limitations of your studies, which is always a good habit of scientists to show what their limitations are. But these limitations are the ones that we often read. This is not to comment on you in a negative way, but I was thinking more broadly, how could we address these limitations and, advance in our sciences? So you mentioned actually in the summary three basic limitations, and perhaps you could have a look at it. The first one deals with environmental factors. So then is the second one more or less with perinatal, postnatal effects, and then of course the genetic background, I should say, within this deletion region. But let's split it into three parts. So what design would you envision to deal with the environmental factors involved?
Hooggeleerde opponent, thank you very much for your kind and indeed creative words. It's a very interesting or important point, I think, that you're referring to, and you're asking me how could we address these limitations that I briefly mentioned in our work going forward. Well, I would start by saying that our work contributes to, so it's not as one of your colleagues pointed out, it leads to more questions than we perhaps previously had, but that's, I think, the deal in science. But it contributes to our understanding of this large outcome uncertainty and this large uncertainty with respect to underlying mechanisms, with respect to phenotypic expression for these individuals. And we are getting better and better, and our studies contribute to that, at discerning aspects that are or are not related to outcome. And by doing that, we limit ourselves in terms of the factors that we can incorporate in our studies. And that is important because, of course, we can think of so many factors that are important in terms of outcome prediction. And with our studies, for example, we've demonstrated, okay, there's no association between ASD and psychosis. However, there is an association between IQ decline and psychosis. So these are important findings, I would say. Nonetheless, they still leave out the important factor of, for example, environment, whereas, we need... Yes.
Let's go to the point. So choose an environmental factor that you think is important and explain to me what design we need.
Yeah. What I believe... Help? Yes. Well, thank you. For example, I think we should include the factor of stress, environmental stress, to include in our studies because, some of our colleagues are already demonstrating in their work that their individuals with 22q may differentially respond to stress than individuals in a general population, and that this may be associated to outcome. How would we do this? Well, we need to do a better job in our phenotyping, in our data collection, which is challenging, and I think we are doing our best already. But as you've read in my work, especially when we want to say something about genotype-phenotype association, we need large sample sizes that are not reasonably collected within one study site, because I would argue within one study site, it's relatively easy to assess everything that you would like to assess. So the drawback of all these very important and necessary large international collaborations is that it's much more difficult to harmonize what we assess. But I would argue that that's important to take into account in a standardized method. And I also think that the smaller studies that are being conducted in specific sites that allow for such assessments pave the way for what should we then be assessing collectively in our larger samples. And I would definitely argue that stress is a very important factor to take into account, given recent studies by our colleagues, but also given the clinical impression that it is. We very often talk about level of impairment on several outcome measures, but I would argue that it is hardly ever, or perhaps maybe even never, the level of difficulty that one has in, for example, cognitive functioning, that in itself leads to distress or psychopathology, but rather it's when stress arises because there's a mismatch between one's individual capabilities and challenges on the one hand and what is being asked of them.
So I of course asked a big question, so I get a big answer. But if you think about stress and you operationalize stress, let's say in some sort of assessment schedule or whatever, could also be cortisol. But then we go back to biology. Mm-hmm. But if you would look at stress, would you kind of think as a sort of continuous variable, or would you dichotomize these data? So if you think about design. Oh, I have 1 minute left.
My initial thinking would be to not dichotomize these variables. I think that especially the experience, well, as you say, stress is a subjective, can be defined in many different ways, but our colleagues have been paving the way for us. There is a very nice method, experience sampling method, I think it's called, where you can, tap into this construct and you can correlate it to more biological measures of, for example, cortisol. And I think that's what we need, an integrative approach of a construct as complicated and important as stress.
Thank you very much. I'm very pleased with your answer. And the other limitations, I think they will require another day to discuss with you. So
I will be happy to.
But thank you very much. I'll hand it back to the rector. Thank you very much.
Thank you very much, Professor Staal, for waking me up to introduce the next opponent. And the next opponent is from the Catholic University of Louvain in Belgium. And we are very happy that you're here, Professor Swillen. Professor Swillen is Professor of Educational Psychology, and she will continue the opposition. Please go ahead.
Thank you, Mr. Chairman. Dear Madam Candidate, dear Ania, everybody, I think I echoes the former comments and feedback on your, excellent work, and I would like to congratulate you, but also your supervisor team. So, my question is about, the first part is about chapter 6, which is on the family within family study, which I think is a very important one. And I would like you to go to page 125, where you present the very nice figure 2, which relates to the within family probe and parent correlation of the full-scale IQ. And so all adults were the ones with a de novo 22q deletion, so no familial deletions. And as we can see, there is, in the majority of them, a kind of a shift or a curve that is, let's say, minus 30 IQ between the biparental IQ and the probant IQ. However, there are also different curves that I see, so different slopes, you could say. And when I look at the lower extreme, so there are about 8 to 10 families, so parents, who have an IQ about between 70 and 85. And if you look to the right, then there is a kind of a subset that seems to have another slope. So my question is, could you comment a little bit on that? What is the possible explanation about these different slopes? A little bit on the opposite, you have another extreme. For instance, one person with a 128 IQ, which is a positive outliner in the 22q. And if you go back to the parental IQ, it's almost a horizontal line. So it's a little bit the same. So what is going on in those subset of families?
Mm-hmm. Hooggeleerde opponent, thank you very much for your kind words, first of all. With your question, you're referring to the correlation that we identified between level of IQ in offspring with the de novo 22q11 1.2 deletion and their non-affected parents. And you're observing something that we've actually discussed amongst ourselves extensively, and that I've thought about and that I would like to investigate further. So I agree with you that there is, if you could call it a trend, that the parents with a lower IQ than expected from the general population may perhaps even be associated with a further decrease in IQ in offspring. And in a previous version, actually, of this figure, we've dichotomized, or we've made a distinction between offspring with intellectual disability and those without. And there, one could actually observe this trend that you're referring to very nicely. So it seems to be there. However, it's our data and our sample size was not sufficient to actually specify different trajectories and quantify them in any way. Nonetheless, that still leaves us with enough to speculate about it. And that is what I'm going to do, because I cannot provide you with an answer based on these data. But we could hypothesize based on what we know, for example, from chapter 7, that there may be something in the genetic background of the parents of individuals with 22q that they share with their offspring, so common genetic background variation, that predisposes them for a low IQ, and that effect may be even further amplified in the individuals where that common genetic variation is such that it predisposes for a lower IQ. And with respect to mechanisms, I don't think we are able to say anything about that yet, but it is an observation that I think is congruent with other observations that we can make, that in the extremes, so in the lower IQ ranges and in the higher IQ ranges, there seems to be an amplification of the effect that we see in the group overall. And that is, I would argue, true for full-scale IQ, but also for other dimensionally assessed phenotypes. I am interested in this, and I would like to investigate these slopes further and see whether it is indeed sort of an amplification effect, as I would hypothesize it is.
Mm-hmm. So thank you. So because of course we are doing research basically because we want to understand more and give us some guidelines of some big important steps for, making a kind of clinical standardized protocols for follow-up. So my question related to this is, what is in fact essential for the clinical follow-up of the cognitive capacities and challenges of these children, adolescents, and adults? And how would you operationalize it into a kind of a clinical setting? So what are key ages? Because you cannot, of course, ask families every 2-3 years to come back to do, let's say, practically, to do both in the child and in the families, this assessment. So what would be a kind of minimal protocol that for following up these cognitive challenges across time? What would be your proposition based on what you know now?
Yeah. There's multiple components that would have to be included in this. I think 3, if I could summarize it like that. The first aspect of what would need to be included in the clinical follow-up of these individuals in terms of cognitive functioning is that the regularity of the assessments is important, because our and your and other studies have demonstrated that it is not a stable construct over the time span, and that we do need to take into consideration the possibility of very different trajectories. Second aspect is that it needs to be comprehensive, so that in an ideal situation, one would want to not only focus on a construct as global as full-scale overall IQ, because we know that in the population of individuals with 22q, there are much bigger discrepancies between different components of cognitive functioning that have very real clinical or real life implications. So comprehensiveness is the second aspect. And the third aspect is when to do it. And of course, that's always a balance between what is reasonable given the societal possibilities and what we would ideally want from a clinical perspective. But I would argue that at the minimum, we would want to see these children and assess them at the ages of transition phases or right before the transition phases. So, of course, that's a little bit dependent on what country you're living in. But for example, in the Netherlands, I would like to see the children around the age of 6 when they start their more formal schooling. Then I would definitely want to see them back before the age of 12, so before they transfer to the next level of their education, and around the age of 16, so before they transition into adulthood. Because we know that these are periods where, first of all, because they're transition phases that may be associated with increased stress and difficulties.
45 minutes. How quick can they go? Don't you agree? But
I do.
Yeah. But hora est, pronounced by the Beagle, means hora est. Time is up. So I think you have got a lot of opportunities to give a very inspiring lecture to the audience. And the committee will now withdraw for further deliberation, and the session will be adjourned. I would like to ask everybody to close this session and go back to the committee room. Thank you.
And the audience may stay in the Senate Hall. We will be back in 10 minutes. Ja, dat lukte niet meer.
Gaat er iets mis?
Nee, het is oké. We are still waiting for the last few people. One more to come. Yes, Professor Knape, you can continue.
Okay. Well, you can see I have my hammer already in my hand, so I hereby reopen this session. Madam Doctoral Candidate, this committee, installed by the Board of Conferral of Doctoral Degrees of Utrecht University, has considered your dissertation and has heard your defense. The committee has decided to confer on you the title of Doctor. I now request the supervisor, Professor Khan, to invest you with the honors in the usual fashion. Professor Khan, please.
Thank you very much, Mr. Rector. By virtue of the authority conferred on us by law, and according to the decision of the Board for the conferral of doctoral degrees present at this session, I hereby confer upon you, Ania Fiksinski, the doctoral degree to which are attached all rights and obligations towards science and society as laid down by law and custom. Do you promise always to perform your duties according to the principles of academic integrity, honestly and scrupulously, critically and transparently, and independently and impartially? I think you have to, yes or no.
You have to put your microphone on, Ania.
Apologies, and my response is I do promise that.
Great, thank you. Then the diploma signed by the rector and supervisors and co-supervisor and bearing the Great Seal of Utrecht University will be presented to you later this year. Thank you. And I now would like to give the word to your co-supervisor, Dr. Forstman, who will give the laudatio.
Thank you, Professor Khan. It is my task to address you, Ania, on behalf of your supervisors, Professor Ann Bassett and Professor René Khan, and through them, actually on behalf of the entire scientific community, to address you for the very first time with your new title. So here goes. It's truly an honor, as well as an immense pleasure, to congratulate you, Dr. Fiksinski, with this important milestone in your life. Laudatio is Latin for praise, and so essentially my job here in a nutshell is to tell your family and friends and colleagues about the hard work and the perseverance and the labor and the blood, sweat and tears and everything you've invested to get where you are today. Yet, I think there's something circular in it. Why? Well, isn't the very fact that you so brilliantly defended your thesis today, and you did it with such success that the entire society now agrees that you've earned your doctorate title, isn't that in and by itself the strongest testimony to your intelligence and to your hard work? I mean, what can I possibly add to that? Of course, I mean, I could tell everyone here that, you know, when you started, you didn't know much about the 22q11 point deletion. You were not medically trained. And yet you managed not only to learn everything there is to know about this particular genetic variant, but much more than that, you acquired such deep understanding of genetics that your recent articles are truly and objectively frontline papers in this domain. Or I could tell everyone here that, you know your languages, your Dutch, Polish, English, Italian, French. Those are the ones I know of. But when you started your PhD, you didn't speak a single word in programming language, in computer language. And yet you managed to learn and master R from scratch and without any formal instruction. I can tell everyone here how quickly you learned to navigate international waters. And you entered the IBBC network. This is the international collaboration around 22q deletion. And in no time, became highly appreciated by all of its members. And, that's probably because you're such a pleasant and social person. But I believe it is also because you were not afraid to put your hands in the mud, and you understood that you just have to work hard. And it's true that the quality control efforts that you did for the consortium data, they continue to be of paramount importance for the work that is continuing to be done by this consortium. And during all of this, you successfully performed your own genetic experiment with your husband, Bruno. And by the way, Bruno, who has single-handedly, you don't know this, Ania, single-handedly coordinated every takeoff and touchdown you made at Schiphol when you were going back and forth between Utrecht and Toronto. But seriously, the point I want to make, he supported you in every move, and you're a real team, and together you've welcomed your adorable daughter, Verena. Among the first two sentences that Verena is starting to use just now, everybody, is Mama PhD. I'm not entirely sure if that is a good sign, but it certainly is endearing. And since we're talking about genetics, may I add that your talent should perhaps not be too surprising, given that you're the child of a mathematician, a physicist, and an accomplished musician, as well as the grandchild of a chemist. And the latter, that's your Babsia, certainly, I know, has been watching today from above, must have been very proud looking at you, how elegantly you waltzed your way through the defense. Ania, ladies and gentlemen, is either an artistic scientist or a scientific artist. I can't decide it, but what I do know is that she didn't come here all the way by walking, but she danced. She danced until today, and she probably will go on dancing. Even all of these feats are definitely laudable. They are still somewhat peripheral to what is my main motivation to sing Dr. Fiksinski's praises today. Dr. Fiksinski quickly understood that medical research requires precise and objective observations. It needs hard work. You need to collect hard data, which allows subsequent analysis to test hypotheses. For all this to be possible, we classify in group observations and we translate them into quantifiable data, basically a series of zeros and ones. And to get there requires tedious and rigorous efforts, discipline and perseverance. And Dr. Fiksinski has demonstrated she has all of that. But it's by no means enough. You see, the real reason why I'm so endlessly grateful for knowing Dr. Fiksinski and so enthusiastic about what she will continue to bring to the world is something else. It is something I consider much more important too. After all, every data point, every single line of code in R, every p-value in your thesis has started with a person. A child with a name and a face, a child with her own history, a mother and a father with hopes and fears and struggles and disappointments and victories and joys. And here's my point, dear friends and colleagues, that unique person, every unique person is what counts most for Dr. Fiksinski. You see, that is the secret to her success. I've come to know her as someone genuinely and deeply interested in every person she meets, their individual story, the complexity of that story, and their well-being. And by the way, this is not her professional attitude. This is just her attitude in life. She's always interested in what truly moves someone and wants to discover the best in each one of us. It's how she is, both in and outside her job, and I've learned a lot from her. Her strive to truly encounter each patient is not a mere footnote in her work ethic. It is the foundation on which she builds both her clinical and her scientific work. And as long as that genuine interest in each person is the bedrock on which the rest is built, my confidence in her future, scientific and beyond, is boundless. So I'm so happy today because the scientific family welcomes a new member who I'm convinced will have a relevant impact as a clinician, as a scientist, or as both. I'm even more thrilled that through this all, I've gained such a dear friend in my life. Dr. Fiksinski, dear Ania, dear Bruno, Verena, parents, extended family and friends, congratulations, and I wish you a joyful day of well-earned celebrations. Thank you.
Thank you very much, Dr. Vosman. Dear Dr. Fiksinski, it's always a great pleasure for me to be one of the first to congratulate you with your new honors. And what should I say after this brilliant laudation? I think it will make you very shy. I think it makes you shy. But what have you done? You've started as a professional dance training, theater training, followed by a bachelor in psychology and linguistics at the University of Utrecht, followed by a clinical health psychology master, and then your studies both in Utrecht and Toronto. You gained several grants and prizes as proof of your excellence. I've seen that you've made many publications, and I've been told a few minutes ago that all the publications have been published already, and your study have led to a better understanding of the neuropsychiatric expression in 22q11ds population, which makes it possible to improve the clinical care and pay important contributions for personalized medicine in this group of patients. This is a very important statement and a very important result with you have to be congratulated. And I'm very happy that you are going to continue your work and are not going to be lost for science, because you will start on a postdoc within a few weeks in the Wilhelmina's Children's Hospital in close cooperation with Professor van Amersfoort, whom we have met this afternoon. I do not only congratulate you, but also, of course, your parents, your mother, Barbara Dutka, and your father, Bogoslav Fiksinski, and we pay our condolences for the fact that only a week ago his mother had passed away. We feel very sorry for you. I always congratulate, of course, your husband Bruno and your little daughter Verena and your brother Mikovaj and David. I also congratulate the paronyms. You were excellent paronyms, although you forgot to unmute the microphone when Ania was addressed during the expression of the formula. I also would like to thank the technicians and the Beadle for her service. And yes, because we are not able to congratulate you, I would like to ask everybody to just clap you in your hand for what you've done.
Thank you.
Having said that, we are going to conclude. Very learned lady, on behalf of the Board of Conferral of Doctoral Degrees of Utrecht University, I congratulate you on the honors which have been bestowed on you. I hereby conclude this session and ask the participants to close the session and go back to the committee room in order to be able to congratulate the supervisory team.
Okay.
So, Ania, my congratulations.
Thank you.
Pleasure for today and success for the future. I hope we will meet in person one time. Yeah.
Yeah. Thank you very much for your help. So, do I now go back to the other meeting?
No, you can leave this session and then you can start party at home.
Okay.
Yeah. I hope the committee members, Professor Knape did it a little bit different from the other chairpersons. So I hoped that everybody could congratulate you, but well, he did it in another way. So I hope the committee members will send you an email or something. Yeah?
Thank you. Yeah.
So pleasure for today. Bye-bye.
Bye. Thank you.
Kunnen we ze niet terughalen?